Dear students, today we are going to learn about nucleic acid called as DNA. What is DNA? DNA is going to be present as the genetic material inside the body of all types of living organism. Except some viruses, DNA is the genetic material inside the body of living organism. In some viruses only, the RNA is called as genetic material. Now, what is DNA? DNA is nucleic acid. DNA is a type of nucleic acid. And the full form of DNA is deoxyribonucleic acid. Deoxyribonucleic acid. Where this DNA is going to be present? This DNA is going to be located inside the nucleus. Inside the nucleus, the DNA is going to be located. And what is the purpose of DNA? DNA will store genetic information. It will store genetic information and it transfers genetic information from one generation to another generation. DNA will play an important role in storing the genetic information, and at the same time it will transfer the genetic information from one generation to another generation. That is, genetic information is going to be transferred from parent to offspring. From parent to offspring in the form of DNA. And they are carried by the help of chromosomes, which are called as vehicles of heredity. Which are called as vehicles of heredity. They will play an important role in the transmission of characters from generation to generation. They will play an important role in transmission of character from generation to generation. Now, how the discovery of DNA came into existence? For the first time, the DNA was discovered by Friedrich Mischer. Is the name of the scientist who discovered acidic substance inside the nucleus. Inside the nucleus, and he named it as nuclein. He named it as nuclein in 1869. After many years of time, there was no proper technical apparatus and descriptions. After many years, nearly about 80 to 90 years, in 1953, with the help of X-ray diffraction studies, two scientists named as James Watson and Francis Crick discovered double helical model of DNA or double helical structure of DNA. Double helical structure of DNA, for which they were awarded Nobel Prize. For which they were awarded Nobel Prize. Till that time, nobody knows the double helical structure of DNA. Double helical structure of DNA came into existence in 1953 after X-ray diffraction studies came into consideration. That time, two scientists, James Watson and Francis Crick, has discovered the double helical model of DNA. After the discovery of the double helical model of DNA, we came to know the following things. What is DNA? What is DNA? DNA is made up of polynucleotide strand. DNA is made up of polynucleotide strand or chain. Polynucleotide strand or chain. DNA is a polymer. Very important. DNA is a polymer. Polymer, what do you mean by polymer? When many monomers are going to be joined together, they will form polymer. Chain of monomers are called as polymer. Chain of monomers are called as polymer, and DNA is going to be consisting of chain of nucleotides. Chain of nucleotide are going to join and form polynucleotide strand. Chain of nucleotide will join and form polynucleotide strand, which is the structure of DNA, basic structure of DNA. What do you mean by polynucleotide strand? But before going to polynucleotide strand, you should understand the structure of nucleotide. The structure of nucleotide, very important. The structure of nucleotide is going to be consisting of deoxyribose sugar, phosphate, deoxyribose sugar, phosphate and nitrogen base. And nitrogen base. This is the structure of nucleotide consisting of phosphate group, sugar group and nitrogen base. When these three things are going to be present together, then this structure is called as nucleotide. Nucleotide is combination of phosphate plus sugar plus nitrogen base. Phosphate plus sugar plus nitrogen base. If you remove phosphate from nucleotide, then this structure is called as nucleoside. Very important question for IP as well as NEET. Nucleoside, sugar plus nitrogen base is called as nucleoside. Phosphate plus sugar plus nitrogen base is called as nucleotide. Now in between the nitrogen base and deoxyribose sugar, glycosidic linkage is present. Glycosidic linkage is present. Very important. And in between deoxyribose sugar and phosphate, phosphoester bond is present. Phosphoester bond is present. These two bonds are very important. The bond which is going to be present between deoxyribose sugar is called as N-glycosidic linkage, and the bond which is going to be present between deoxyribose sugar and phosphate is called as phosphoester bond. Now what is the chemical formula of deoxyribose sugar? This question will always come for NEET, very important. Chemical formula of deoxyribose sugar and structure of deoxyribose sugar. It is very important for biochemistry also, and this question will come for biology also. So the chemical formula of deoxyribose sugar is C5H10O4. C5H10O4 is the chemical formula of deoxyribose sugar. Now what are the four types of nitrogen bases which are going to be present inside the DNA? The DNA is going to contain four types of nitrogen bases: adenine, guanine, thymine and cytosine.